Good morning, everyone. I hope we are finally able to start. And today we have Tsigen Shah from the Max Planck Institute of Psycholinguistics, which is presenting a very interesting talk that has been recently published in Science Advances about genetic architecture for the white matter connectome in the human brain. So, Xinqiang, if the screen is yours, you can go ahead.
And white matter connectivity is also accompanied with dynamic changes during neurodevelopment and aging period. So this figure illustrates the alterations of spatial distribution of hub regions, which reflect the importance of given region within the white matter connectivity network. So in this figure, each dot represents a given region, and each yellow edge represents what matter connection between a pair region. So we can see the hub regions primarily distributed in the anterior part of the human brain during adolescence, while moving towards the posterior part of the brain over time. So speaking of DTI, I guess many people might remember some well-known DTI mirrors. For example, FA, AD, RD, and MD. So what's the difference between these white matter microstructure mirrors and white matter connectivity that we introduced today? So let me talk about the difference. I think the most important knowledge background is that diffusion rate of the water molecules in each direction within the axon is different. So based on this basic principle, DTI can model the diffusion of water molecules in 3 directions, including lambda 1, lambda 2, and lambda 3. So FA represents the degree of anisotropy different from isotropy, and AD represents the diffusivity in a primary diffusion direction, and RD represents the diffusivity independent of the primary direction, that is lambda 2 and lambda 3. And MD represents the average diffusivity across all of the directions. And these mirrors can capture the local integrity of what matter microstructure, and also can capture some underlying biological features. So for example, FA has been reported to be associated with the axon density, axon count, and myelin content. And in contrast to the white matter microstructure, white matter connectivity can be constructed by the fiber tractography. And this technology is based on another very basic principle, that is the diffusion tensor major engine vector is supposed to be parallel to the local white matter tracks. And we can reconstruct the fiber tracks by linking the voxels that has similar orientations, bulk continent streamlines between the start point and end point. So for example, if we start from this point and then move along the voxels with similar directions, and then we can very easily get the streamline between 2 points. So previous GWAS analysis have identified 100 genetic loci associated with different aspects of white matter microstructure. And significant genetic correlation have been also observed between these microstructure mirrors and brain disorders such as stroke, major depressive disorders, and glioma. However, the genetic architecture of the white matter connectivity and its association with brain disorders are still unknown. So before I talk about some genetic stuff or biological stuff, I think I need to clarify 2 concepts. So what is SNP, or what is genotype? So SNP is a variation at a specific position in the genome. So for example, at this specific position, G could appear in most of the individuals. Well, this position can be also occupied by T in a minority of individuals. So that is to say, at this specific position, there is an SNP, and G and T can be both considered as alleles at this specific position. And previous large-scale SNP array technology has sequenced millions of SNPs across the genome from different species, humans or non-humans, or some alienwares, of course. And this study also report some SNPs can affect its gene expression. So for example, this figure is a very good example illustrating the association between the SNP and the expression level of the closest gene. So for example, this SNP is located within this gene. And this figure, we can see the green, the blue dots represents the gene with GG, and the gray dots represent the genes with AG, and red dots represent the gene with AA. So we can see that in this figure, the genes with GG tend to show higher expression than the genes with AG, and the genes with AG tend to show higher expression than the genes with AA. So that means the more G this gene has, the higher the expression this gene will be. So in this study, we carry out the brain-wide tractography on 30,000 individuals from the Biobank. Our aim is to first estimate the heritability and then perform the multivariate GWAS across these heritable traits. And regarding these identified genetic variants associated with what matters connectome, we also wanted to do some functional annotations to identify some relevant gene sets of biological processes and functions associated with what matter connectome formation. And then we also map the polygenetic effect of the brain disorders of the structure connectome, including a set of psychiatric disorders and neurological disorders such as autism, schizophrenia, bipolar disorder, and ADHD, ALS, and Alzheimer's disease, together with handedness. And finally, we take the language functions as an example to illustrate how we inquire and understand the genetic mechanisms of the what matter connectivity that is associated with a given function, cognitive functions, by looking up our identified genetic GWAS summary statistics. So first, we try to do the tractography on each of the individuals, followed by constructing a what matter connectivity matrix. And regarding this connectivity matrix, it requires two definitions, including node definition and edge definition. For node definition, we mapped the AAL atlas from common space to native space, and a total of 90 regions has been defined for each individual. And for the edge definition, we counted the number of streamlines between each pair of the region while adjusting for the brain size of the volumes of the regions connected. And this process roughly takes 4 months on 6 cluster nodes, and this process also used 110 gigabytes. And we analyzed some brain matters at 2 levels, including connectivity level and node level. At the connectivity level, so this figure is a very good example to show how we get the connectivity level data. So these are the regions of our interest. So we can see there are a lot of streamlines linking this parent region. And we counted the number of streamlines between 2 regions while adjusting for the brain size of the volumes of these regions to define the connectivity. And at the nodal level, we counted the degree centrality for this region by summing the connectivity linking a given region. For example, this green region has 4 connectivity, and the degree centrality of this region is 0.t plus 0.2 plus 0.4 plus 0.3, that is 1.1. And then we did a sample and genetic quality control for the follow-up genetic analysis. For the sample quality control, we excluded outliers based on heterogeneity, genotype meaningless, and then we'll also exclude some subjects with mismatch of self-reported and genetically inferred sex. And we only include the subjects with what British and census tree, just trying to improve the genetic homogeneity. And in this study, we also included the subjects with relatedness of kinship coefficients higher than 0.04, just wanted to exclude these subjects with relatedness. And for the genetic quality control, we only included the common genetic variance with minor allele frequency higher than 1% and infer score higher than 0.7 and Hardy-Weinberg equilibrium p-value higher than 10⁻⁷. And then we extracted the nodal level measures and edge level measures for each individual. So finally, we included 30,000 participants with 14,000 males and 16,000 females. And then we performed the S&P based heritability. So if you wanted to divide the variance of a given trait, we can get 2 components. So first, a component can be explained by the genetic factors, and another component can be explained by the environmental and the trans factors. So heritability is to estimate the proportion of the given trait that can be explained by all of the common genetic variants across the genome. And the genetic relatedness between individuals can be measured from the genome-wide data and linked to the trait variability and further estimate the heritability. So specifically, after removing the current relatedness, we constructed a genetic relationship matrix between each pair of individuals and calculate genome-wide restricted maximum likelihood. And for the multivariate GWAS analysis, we first tested the union-variant associations separated for each SNP and a heritable trait. And then for each SNP, we'll convert the p-value to z-score and then use Mahalanobis to compute the multivariate association statistics. And finally, we'll also identify some important regions or connectivity that make the greatest contribution to the overall multivariate associations. So this is to just want to identify some important regions or connectivity driving the multivariate association signals. And in this genetic analysis, we also included a set of covariance to control for the confounding factors. So these covariance include age, non-linear age, and genetic components, as well as the center, genome batch, and sex. So here are the main results of our heritability analysis. So first, to estimate the heritability separated for regional degree centrality and white matter connectivity. So this figure illustrates the brain-wide heritability of all of the regional degree centralities, with red representing the regions with higher heritability. So we can see the regions with higher heritability primarily involved in the dorsolateral prefrontal cortex, superior temporal cortex, and posterior cortex, as well as medial temporal cortex. And then after buffering correction, we found all of the regional degree centrality show significantly heritable. And for the white matter connectivity, in this figure, each edge represents the connectivity between each pair of region, with red representing the connectivity with higher heritability. And after buffering correction, we found there are 851 structural connectivity, showing significant heritability. And interestingly, we found the heritability of interhemispheric connectivity showed relatively low than the heritabilities of intrahemispheric connectivities. And then we perform the multivariate GWAS analysis separated for regional degree, centrality, and wet matter connectivity. So this is the Manhattan plot illustrating the multivariate GWAS results. So we can see each color represents one chromosome, and each dot represents a SNP. And for the regional degree centrality, we found 140 lead SNP in 117 genetic loci associated with regional degree centrality. And for the wet matter connectivity, we found 211 significant lead SNP within this 166 genomic loci. And except for chromosome 21, we found each chromosome has at least one significant genetic loci associated with either degree centrality or wet matter connectivity. And then we try to identify some important regions of connectivity that drive the overall multivariate associations. So in this figure, we can see this figure illustrates the extent to which the regions contribute to the overall multivariate associations. And with red representing the greater contribution. So we can see the regions with greater contribution to the overall multivariate associations primarily involved in dorsolateral prefrontal cortex and medial prefrontal cortex and putamen. For the connectivity multivariate GWAS, we found connectivity between precuneus, calcarine, and post-central precentral cortex drive the overall multivariate associations. And then we try to map these SNP associated with wet matter connectivity or wet matter connectome to genes and try to identify some relevant biological pathways. So before I talk about this process, I think we need to clarify the process from DNA to protein. So how does DNA plays a crucial role in the follow-up biological pathways or protein level? So as we know, DNA is the genetic material in our cells. And DNA as the commander can't directly involve the biological pathways or processes. So how does DNA plays the important role in our daily life? And classical molecular hypothesis reported that it needs 3 steps. So the first step is DNA can be transcribed to messenger RNA. And this messenger RNA can be translated to amino acid chain. And this chain can be folded to protein through processing in organelles. So I think this process is very abstract for the persons without biological background. So I have a video to show this process very intuitively. So let's check it out, this video. Yeah. So hold on a second. So this is our cell. There's a structure called nucleus, which contains the genome. And the human genome has 23 pairs of chromosomes. And each chromosome contains DNA which tightly package the round histones. Within DNA are the section called genes. When a gene is switched on, RNA polymerase attaches the start of the gene to make a strand of message RNA. And this process is called transcription. Before mRNA can be used as a template for producing protein, it needs to be processed. And then the mRNA moves into the cytoplasm. The ribosome binds the mRNA and reads the codes of the message RNA to produce a chain of amino acids. So the transfer RNA carries amino acids to the ribosome, and this is added to a growing chain of amino acids. And this process is called translation. So once the last amino acid has been added, the chain folds to a complex three-dimensional shape to form the protein. So this is a protein. So that's how DNA plays a crucial role in the protein or biological functions. So let's go back to the topic. So then we use Magma toolbox to perform the gene-based association analysis, and the resulting gene-based p-value can be used to test the gene set enrichment analysis and further identify some relevant biological pathways. So for the degree centrality GWAS, we found 61 significant gene sets with neurogenesis and neurodifferentiation shows the strongest enrichment. And for the white matter connectivity multivariate GWAS, we found 72 significant gene sets with axon-related processes showing the strongest enrichment. I think these results are very consistent with previous studies because our matters are derived from white matter, and white matter are enriched in some astrocytes and axons, as well as microglia. And then these gene-based p-value can be also used to test the differential expression pattern across neurodevelopmental stages by using brain span datasets. And we found the genes associated with both network degree and white matter connectivity shows differential expression during the entire neurodevelopmental stage, spanning from early prenatal brain to late prenatal stages, as well as from 9 weeks to 26 weeks in the prenatal brain. And this gene-based p-value can be also used to test the enrichment of the cell types based on the single cell gene expression data. And for the cross age groups, we found genes associated with both network degree and white matter connectivity were upregulated in astrocytes. And broken down by age, we found the genes associated with degree centrality showed upregulation in microglia at 10 weeks, and astrocyte at 19 weeks, and neurons and astrocyte at 26 weeks. And for the genes associated with white matter connectivity, we found these genes showed upregulation in stem cells at 10 weeks, and neurons at 16 weeks, and astrocytes and gamma neurons at 26 weeks. So as we know, white matter connectivity is also accompanied by the variety of neuropsychiatric disorders. So this figure illustrates the shared white matter connectivity alterations across these brain disorders. So you can see this figure, dark color represents the regions with white matter connectivity alterations across brain disorders. And if we divide these regions by network analysis, we can find these regions, these patterns is quite similar to the pattern where hub regions distribute. That suggests the regions with dense white matter connectivity tend to be more vulnerable to brain disorders. And then we try to generate the population risk score to map the genetic effect of the brain disorders on the white matter connectome. So population risk score is a single score that can capture the genome-wide risk for a given disorders. So that is to say, the higher the population risk score is, the higher tendency this individual express the clinical characteristics for a given disorder. And then we use the canonical correlation model to construct a multivariate association between the regional degree and the population risk score for each disorder. And then we can derive the loadings of the population risk for each disorder on the regional degree centrality. And the brain regions with higher loading can be used to generate a single mask. And this mask can be used as input in the neuroscience data sets to derive a set of cognitive annotations. For example, if we input this mask into the neuroscience data sets, we can generate a coactivation map and then derive the sets of cognitive annotations. For example, this brain region could be associated with inhibition function and social interaction function and episodic memory. And for the multivariate association analysis, we found significant association signal between the degree centrality and the population risk for each disorder. So this result suggesting the population risk for each disorder might partially reflected in the white matter connectome. And this figure illustrates the loadings of the population risk for each disorder on the regional degree centrality. And for the psychiatric disorders, we can see the loadings of the population risk for each disorder or widely distributed across the brain, suggesting the higher population risk for psychiatric disorders was associated with lower regional connectivity across the brain region. And for the neurological disorders, we found that loadings, the negative loadings were widely distributed across the brain regions, suggesting the higher population risk for neurological disorders was associated with higher regional connectivity in the majority of brain regions. And for the handedness, we found some regions were associated with language-related functions, such as Broca area, superior temporal cortex, and visual cortex. And finally, let me take the language function as an example to illustrate how we inquiry and understand the genetic mechanisms underlying the white matter connectivity that are important for a given cognitive function. So for example, there are 4 regions has been reported to be associated with language function, including Broca 44 and 45, and superior temporal cortex and medial temporal cortex. And then we extracted these 4 regions from our AAL atlas to investigate the genetic correlates of the white matter connectivity underlying these 4 regions. So this table illustrates statistical values extracted from the univariate association analysis, with orange showing the significant association signals. So we can see there are 4 lead SNP showing significant associations with at least 3 language-related connectivity. And if we map these lead SNPs to genes, and we can take a look at which subset of genes was associated with a specific language connectivity. So these GWAS summary statistics can also provide insights into the genetic variants, genes, or biological pathways associated with a given cognitive functions that we're interested in. And in the gene set enrichment analysis, as I just mentioned, we found the genes associated with degree centrality was enriched in the neural differentiation neurogenesis processes, while the genes associated with white matter connectivity might involve the axon pruning. And SEMAS3A is a top gene in the gene space association analysis. And this gene also plays a crucial role in both neurodifferentiation and axon-related process. So we could hypothesize that neurodifferentiation might be very important for the overall white matter connectome formation, while axon pruning might shape the precise white matter connectivity. And in the neurodevelopmental stage analysis, we found the genes associated with degree centrality and white matter connectivity showed the differential expression during the entire neurodevelopment. So this figure illustrates the trajectory of white matter network formation during the neurodevelopment. So we can see the radial fiber tracks extend to multiple directions at multiple scales during the entire neurodevelopment. So I hypothesize that these white matter related genes could take an active part in the overall multi-model white matter connectome formation. And let me use the 3 sentences to make a conclusion. So our GWAS analysis identified 325 lead SNPs across the genome, which 80% of the SNP hasn't been previously reported in a study. And the enrichment analysis implicated neurodevelopmental processes, including neurogenesis, neurodifferentiation, and axon-related processes, as well as prenatal brain expression, especially in the astrocyte, microglia, and neurons. And polygenic score for neuropsychiatric disorders show significant multivariate associations with structural connectivity, and each implicating distinct sets of the brain regions with trait-relevant functional profiles. And finally, I want to thank my mentor, Clyde and Simon, and other colleagues in border universities and University of Oslo. And I also wanted to thank the UK Biobank to provide this large-scale neuroimaging genetic data set to let this dream come true. Yeah. And also thank the Max Planck Society to support this study. So thank you so much for your attention.